So the last time we were talking about the structure of common nucleotides. Now we're going to take a deeper dive into the way these molecules function. So in nucleotides, the pentose ring is attached to the nucleobase via a glycosidic bond. So this is at the anomeric carbon. So that position is formed at the N1 in pyrimidines and at the N9 in purines. And this bond is very stable towards hydrolysis, especially in pyrimidines. So that bond cleavage is catalyzed by acid. If I have relatively free rotation then around the N-glycosidic bond, I can actually denote the torsion angle by the symbol C. And if I define the sequence of atoms, then I can talk about this angle being 0 corresponding to a syn conformation versus an angle near 180 corresponding to an anti-conformation. And that anti-conformation is typically what we find in bDNA. So if I take a look here, I can actually see the rotation of the base as I've numbered the carbons to be able to see that corresponding structure. So if you take a look at syn versus anti, it gives you a comparison. It's kind of like a swivel on a stick. If I think about the tautomerism of nucleobases, then I have these tautomers that are structural isomers that differ simply in location of the protons. So keto-enol tautomerism is very common in ketones. You study that in organic chemistry. Lactam-lactam tautomerism occurs in some heterocycles. But the key here is that both tautomers exist in solution, but the lactam forms are predominant at a neutral pH. So this is your lactam versus your lactim, or a double lactim in terms of the position of those double bonds. Absorption of UV light at 250 to 270 nanometers is due to the pi star electronic transitions. And these excited states of nucleobases tend to decay very rapidly without radiation. So you can actually see this photoprotection of genetic material. Like our genes are relatively stable to light. And you don't really see fluorescence from nucleic acids. This gives you an example of the wavelength of light and the molar extinction coefficient for nucleotides. And this is some useful nomenclature that you need to be familiar with in terms of what the base is, the nucleoside, the nucleotide, and then the nucleic acid that they're found in. So if we think about the structures, names, and symbols, both the 2-letter and the 4-letter codes, it's useful to know those because it's going to give us some shorthand tools to be able to identify deoxyribonucleotides. You also need to know the ribonucleotides with their 1-letter and 3-letter codes. If we want to think for a moment about minor nucleosides in DNA, it's important that we think about the fact that modification is actually done after DNA synthesis. So the 5-methylcytosine is common in eukaryotes. It's also found in bacteria. The N6-methyladenosine is common in bacteria, but is not found in eukaryotes. And we really want to think of these in terms of epigenetic markers. So this is a way to mark our own DNA so that the cells can degrade foreign DNA, for example, in prokaryotes. It's also a way to mark which genes should be active in eukaryotes. And it's this question of, can the environment turn genes on and off in some type of inheritable way? So here are some examples: the 5-methylcytidine, the N6-methyladenosine, the N2-methylguanosine, and the 5-hydroxymethylcytidine. Inosine, sometimes found in the wobble position of the anticodon in tRNA. This is made by deaminating adenosine. It actually provides a richer genetic code. Pseudouridine is found widely in tRNA and rRNA. It's most common in eukaryotes, but it's also found in eubacteria. It's made from uridine by enzymatic isomerization after the RNA synthesis, and it actually helps to stabilize the structure of tRNA, and it may help in the folding of rRNA. You can see the structures of those here. So the inosine, the pseudouridine, the 7-methylguanosine, and the 4-thiouridine. So when we think about polynucleotides, we're talking about how these nucleotides are formed via our nucleotide. So if we have covalent bonds via the phosphodiester linkages, so essentially that's phosphate, sugar, phosphate, sugar, phosphate, sugar, That's the negatively charged backbone. It's fairly stable. For example, they're able to isolate DNA from mammoths, where the hydrolysis can be accelerated by enzymes like DNase. And the RNA backbone is less stable. So in water, RNA only lasts for a few years. In cells, mRNA is degraded in a few hours. So these are linear polymers. There's really no branching or cross-linking. And there's directionality. So the 5' end is different from the 3' end, and we tend to read from 5 to 3'. So this gives us an example of what that looks like. I can see my 5' end with my phosphate. Notice it's 5 because of the carbon, just like this would be my 4 carbon, and then attached to my 3' carbon is another phosphate, another sugar, another phosphate, another sugar, remembering that my base is attached at the anomeric position. And again, I'm going to see hydrogen bonding between my nucleobases. That's all for now. We'll pick it up again next time.